Great, thank you so much, Kelsey. So let's go to our first speaker, which is Dr. Daniel Wang Prasad. I hope I did this correct, which I know I didn't, but sorry, Daniel. So a little bit about Daniel. Daniel is an interdisciplinary marine scientist with broad research interests including coral reefs, photosynthesis, and bio-inspired design. He is currently working at the Department of Nanoengineering at University of California, San Diego, and Scripps Institution of Oceanography, where he's developing new 3D bioprinting approaches for synthetic coral-algal symbiosis, coral reef restoration, and algal biotechnology. He was a Marie Curie Fellow at the Bio-Inspired Photonics Group at the Department of Chemistry at the University of Cambridge, where he worked on optimizing microalgal cultivation using coral-inspired materials. He received his PhD from the University of Technology from Sydney, where he worked on the biooptics of corals. And with that, let's move to Daniel's presentation. So Daniel, go ahead.
Yeah, thank you very much for the introduction, Katerina and Kelsey, for organizing this, and also thanks to my colleague Lucas for suggesting me to speak. So I'm gonna start here. Okay, you can see the presentation view. Great. So I'm gonna talk today about coral-inspired living photobioreactors, which is my postdoc work that I did for the last 3 years in collaboration with University of Cambridge in the Vignolini lab, and also at Scripps in the marine biology department. And so I'm gonna start with a thought experiment. So imagine you are round, photosynthetic, and there are many of you. And now at first approximation, your main aim is to optimize light absorption for all units. The question is, how would you arrange yourself? Now, let's say this is you, and light is incident from the top. And you can, of course, arrange yourself in a compact configuration. But the problem is you have heterogeneous light distribution. So in a purely light absorbing medium, you have exponential light attenuation, and you have too much light at the top and too little light for the cells that are deeper. So you could also stretch out. But here the problem is now that the space limited, right? So you occupy a lot of space. You could also arrange yourself randomly, but then of course you have to be lucky. Alternatively, you could start mixing. And in fact, this is what most current algal cultivation techniques rely on. So if you look into commercial production of microalgae, you have some low-cost systems here that are raceway ponds where the water is circulated, and this ensures that all cells receive the same amount of light. You have these tubular photobioreactors down here, where water runs through, and as well as these small lab-scale photobioreactors. They all rely on turbulent mixing. Now, mixing is costly, and so we wanted to optimize this. And now, alternatively, what about if we are able to control photon propagation? Let's say we're able to scatter light in a way that each cell, each photosynthetic unit, receives the same amount of light. So you have a homogeneous light distribution. Does such a strategy exist in nature? And basically, the idea is that corals, in fact, employ such mechanisms. So corals live in a very space-competitive environment. So a tropical coral reef, it's very dense. There's high competition for space and resources. So and corals are attached to the benthos, right? So they can't move. And so over millions of years, they have evolved to become a highly optimized light absorbing system. And I have studied the optical properties of corals for many years and started in my PhD. And basically, it shows that they're very good at using light. And today I want to introduce some of these tricks that corals use. So first part of the talk is about coral light management. In the second part of the talk, we talk about the application and the development of these bionic 3D printed corals. Now, because this is, I guess, a very multidisciplinary audience, here's a brief introduction to what a coral is. So at the heart of the coral, you have the symbiosis between the animal host, which is basically an invertebrate, cousin of a jellyfish, and you have microalgae. So these are the brown little dots here that generally belong to the genus Cymodinium, but now they changed it, and it's this family called Cymodiniaceae, which is very difficult to pronounce. Anyhow, so the interaction relies on that these algae photosynthesize, and they then transfer that fixed carbon to the animal host. In return, the animal host provides what we call junk food, so some sort of nutrients that the algae needs, but it also optimizes the physical-chemical microenvironment, and specifically the light regime. This is what I'm going to talk a bit about now. So one of the things that's quite striking, you can see visually, is that the coral actually scatters light strongly. So this here is an example of an incident laser beam, a red light. That's the width of the beam, and then you can see how the light is scattered across the surface of the coral. And using small fiber optic microsensors, we can experimentally measure the spatial distribution of that scattered light. And without going into too much detail here, it shows that the coral tissue is strongly light scattering. Now, this light is also of relevance for photosynthesis. So if you repeat the same experiment, but you exchange this fiber optic microsensor with an oxygen microsensor, you can turn off and turn on the incident laser beam and measure the contribution of the laterally scattered light to oxygen evolution locally with such a microsensor. And it strongly affects photosynthesis. So how much does tissue scatter light? To do this, there's of course various techniques to estimate the optical properties of soft tissues. What I had started to look into was to use optical coherence tomography, which is basically an optical analog to ultrasound. And it uses near-infrared light, which penetrates the tissue nicely, especially in such coral tissues where there's a minimal absorption in this optical window there. And what you can see here is a custom-made OCT system and a big chunk of coral, and it's being imaged here. And the way the data looks is a bit like this. So this here is a polyp that has been imaged. That's a USB camera image. If you calibrate the OCT system, you can also use the signal attenuation of the A-scan, so of the vertical attenuation of the OCT signal to calculate some optical properties. So we have developed a model that calculates the anisotropy of scattering from the local peak of reflectivity here, and the attenuation yields the scattering coefficient. If you have determined some of these optical properties, then of course you can also run a light simulation. So in this case, we developed a Monte Carlo simulation, which is a probability distribution model where lots of photons are emitted. And the nice thing is about this, you can configure the architecture at your desire. So what we have here is an earlier model that we developed, where you have a simple two-layer system. You have this tissue here on top, that's the outer layer, and then you have the skeleton underneath. And then in color coding, you see diffluence rate, so the distribution of light when you have an incident collimated beam that is emitted from the top here. And then if we plot the attenuation as a function of z, what you can see is that as we approach the coral surface, you already see the strong enhancement in the fluence rate in top of the water. And then once you have entered the coral tissue, you reach the strong subsurface maximum. And then you have rapid attenuation. But once you reach the skeleton, this attenuation is reduced. Now, what this shows is that coral tissue scattering approximately doubles the incident irradiance. Now, what are the sources of light scattering in coral tissue? One aspect that's rather interesting are these red dots here. So that's one coral polyp, a couple of millimeters in size, and we look at this OCT scan. And if you see right on the top, you have these red dots. These are green fluorescent protein like pigment granules. So these are high aggregations of GFP, and they strongly scatter light. We've looked into this in a bit more detail, looking at the granule structure. Here, if you shine blue light on it, you can nicely see, visualize them. And if you do some spectral imaging on it, you can see, of course, this. So here in green is where the granule is, and in red, that's the background tissue. You see the absorption, of course, but then you can see this nice green emission. But you also see this very broadband scattering that extends into the 700 nanometer range. And if you do some experiments where we can upregulate the production of GFP in a coral, we can see that the local scalar irradiance, so the fluence rate on the tissue surface, is strongly enhanced when you have more GFP. So by having corals here with upregulated GFP, you can easily double the irradiance. Now let's talk about the skeleton a bit. So we know now tissue scatters light, and what about the skeleton? A skeleton, our hypothesis is this. So if you have, for instance, light that's incident at an oblique angle here, 45 degrees, now light will penetrate through the tissue and is rapidly attenuated and absorbed. But then once it reaches the skeleton, it actually travels quite far. So if you have algae that are situated on the otherwise shaded side, they would receive still sufficient light to photosynthesize. If you run the same simulation, but now you replace the skeleton with a solid mass of living tissue, you can see how rapidly the light attenuates. So the idea really is here that the skeleton acts as a light distributor. There's many tricks actually that corals employ, so I'm just going over a few. So, and the thing is, a coral is not a coral, so there's lots of diversity, and different corals employ different strategies. But here's a cool example of this coral called Pavona, and basically what you see, the brown stripes are where you have high density of algae. The white areas are where you have skeletal extrusions. So basically, light is incident on a skeleton and then gets scattered and distributed into these dense alga layers. So they work with the separation of a strongly scattering and a strongly absorbing medium. Now, another cool thing is that corals are actually dynamically responsive to light intensity. So if you have a coral here, that's an OCT scan, and you see in darkness, the polyps are extended. Now we shine highlight on it, it fully contracts. And if you run an OCT scan on it, and you follow this polyp contraction behavior, you can see how it rapidly contracts, but you also see that the signal intensity increases. So with that, you have an increased scattering. So basically, by changing the tissue arrangement, the coral can control how much light is incident and is being absorbed. So in a way, in the highlight, it kind of protects itself. And the lower light, it expands and optimizes tissue light absorption for photosynthesis. If you look at the point spread function of an incident laser beam, you can also visualize this here. In an expanded state, more light enters the tissue, the skeleton gets laterally distributed here, while in a contracted state, more light is locally backscattered. Okay, so this is an overview of some of the tricks that corals can do. And we have light scattering in tissue and skeleton. We have these GFP granules that can be photoprotective. We have the skeletal architecture, the shape that can optimize photopropagation. And this often we see this distribution of scattering and absorbing elements, so that the algae can receive indirect diffuse light. And then there's, of course, this dynamic modulation aspect as well. Now, my hypothesis was that, well, if they're so good at using light, and people are trying to cultivate microalgae, for bioenergy and bioproducts and so on, and they have lots of troubles actually with optimizing light regulation, can we use some of these strategies? Can we solve some of the key limitations of microalgae cultivation by using a bi-inspired approach? And so my idea was then to engineer a biomimetic coral and then 3D bioprint it, basically. And so my wish list was that I can control the morphology, the microscale architecture. I can control the mechanical properties and tune the optical properties, and it has to be cell compatible. And if you look into the field, of course, and 3D bioprinting has rapidly advanced. It's been lots of work done in medical sciences, but also using bacteria more recently, and some preliminary work had been done with microalgae, but on much larger scale. So I thought that would be a good approach and technique that could fulfill my wish list here. And so the idea was to create a coral host microhabitat mimic to fuel dense microalgae growth. And some of the things, some of the workflow basically involved first to define the input architecture. For the first project, we used some of these optical coherence tomography scans that we then had to segment, binarize and get the surface out, get the skeletal architecture out, develop a CAD model, and then this CAD model could be used for 3D bioprinting. And the important thing was comparison to, using micro CT and so on. We're working with soft tissue. So OCT was very suitable because it had very good spatial resolution and also it's non-invasive. So you could do it live imaging using different tissue architectures under different actinic light regimes, for instance. So there was a useful imaging technique there. And then for the bioprinting approach, we used light-assisted 3D bioprinting. So this relies on photopolymerization. So a brief overview on the development of the bioink. So you have a monomer. In our case, we used gelatin methacrylate, and you add your cells, which are different strains of microalgae cells. And then you can, of course, have different things in there. You have soluble compounds, nutrients, so on. You can have controlled release particles as well. And then you add a photoinitiator and shine light on it. So, and then you have your cross-linked biopolymer. And the technique we have in our lab is based on a digital mask, where we use a digital micromirror device. So in this case here, we have photopolymerization at 4 or 5 nanometer. And so this light is projected onto this DMD chip. And then the DMD chip can be either an off or on position. And when you feed, so the digital mask is basically sliced in Z. So for each Z layer, you have one mask. And then basically start by printing from the bottom and move upwards. And so that's an example. You know, the white areas are on, the black areas are off. And when the mirror is in off position, it directs towards a light absorber. And if it's on, it goes towards the projection lenses. So you have some collimating optics here, and that shines onto your objective stage, where you have your pre-polymer. So the pre-polymer basically is all of this bio-ink mixture, where you have your cells randomly distributed. And then you create patterns, right? And then you load the next mask and the motor. So we have a motor in here. The motor moves, and by that you have some kind of continuous printing. So very smooth structures, and I would say less artifacts between layers. And yeah, also quite good spatial resolution. So down to yeah, less than 10 micrometer. Yeah, so that's the principle. And this here is now an example. That's a coral called Stylophora pistillata, which is sort of the lab rat for coral reef scientists. And so we thought it would be a good thing to bioprint. And if you look at it, it's a branching coral. If you look up close, you have this aragonite skeleton here. And on top of it sits this living tissue with the polyps expanded here in this case. And so here is our OCT scan of the skeleton. Here's the living tissue. And then on top is the 3D print, the single layer, and then a double layer with the printed skeleton, and on top the living tissue containing the Cymboidinium microalgae. We printed all sorts of different architectures. This here is Pavona that you've seen previously. Here's an image of the skeletal architecture. That's the OCT scan. And here, the 3D printed mimic. The same here, that's Passalapora. So I'm going through all sorts of different calls to show you a bit the microstructural diversity, and also the printed replica. Yeah, this is the tissue architecture here of Pavona. The call I showed you before with this nice separation of absorbers and scatterers, and OCT scan and 3D printed mimic. Same here for Vitis flexosa, 3D printed mimic. And so after we had done all these architectures, we were interested in, you know, creating a more simplified bionic coral with the primary aim to optimize light propagation. So you know, started off looking at Stylophora and looking at the skeletal architecture, looking at the key features, and trying to optimize that. So really, with some simple drawings and you know, understanding a bit better what's going on in terms of the skeletal optical properties and light distribution. And so we came up with this 3D printed simplified replica here, which still has the synostal spines and some of the key features. And that's our rendering of our bionic coral here. And then we did experiments with that. And then we, you know, in order to allow easier fabrication, we actually changed this to a more simplified version where you have a cup-shaped corallite architecture that's doped with CNC here at the surface, and so cellulose nanocrystals. And we'll talk about this in a second. That's kind of the final design that we ended up using. So about you know, tuning light scattering in these scaffolds, we started off using silicon dioxide particles, which kind of worked, but not really, because the problem was that the sinking speed was too fast. So we had poor optical control. So you had kind of a gradient during printing. And then we turned on to cellulose nanocrystals, which, of course, in our lab, Silva Vignolini's lab, a lot of people work with. So, there we have a good understanding about their scattering properties, and so cellulose is abundant, is cheap, and can be nicely functionalized to give desired scattering response. Additionally, also it induces some sort of mechanical strength to the hydrogel, which was also beneficial. So we worked on using CNC, So we started a bit late, right? So I'm not sure how much time I have left. Can someone give me an update? So yes, did we start at 3:10?
I would say 5 more minutes, Daniel, if you can.
Perfect. Perfect. Okay, I'll go fast. So here we have kind of the slide strategy scheme, and you would have this, strong light attenuation in the coral. The idea was to reverse that and create an enhanced contribution of scattered light the deeper you travel into the light-absorbing tissue, so you create an inverse light gradient. And in order to do this, we did a lot of bio-ink adjustments, and using different techniques such as OCT, but also using the optical goniometer and integrating sphere to characterize the optical properties of our hydrogel scaffolds, and then tuning them. Here's an example of the skeleton, 0% CNC, and that's 7% CNC. So you can see the scattering visible by naked eye. And then, of course, we can measure it with a small fiber optic microsensor here that measures the fluence rate. So it has a small tip size of 80 micrometer. That's on a coral, but we did this in these hydrogels as well. And then you can see how the addition of CNC enhances the fluence rate as you go deeper into the coral tissue or in the hydrogel scaffold. That's a 3D simulation of the temporal point spread function. So you have incident light here, collimated pencil beam, and we simulated this for purely light absorbing medium, a simple gelatin methacrylate scaffold, and our optimized bionic coral. And you can see kind of the light distribution after 0.5 nanoseconds, after 3 nanoseconds, and 4.5 nanoseconds. Now, the important thing to note is that after 4.5 nanoseconds, there's no more light in the pure light absorbing medium. This has exited the object. In JALMA, there's still some there, but not very much. But once you work with our optimized bionic coral, there's lots of scattering, so you have higher photon residence time and therefore higher chance of light absorption by the light absorbing tissue. Now, if you do these scalar radiance measurements in hydrogel scaffold with a normal JAMA hydrogel and then the bionic coral, and you encapsulate the same density of microalgae, you can see this inverse light gradient. So that's over wavelength here, you can see the main chlorophyll A absorption peaks. They're like 420 and 675. You can see there's almost minimal absorption, and then you have this actually enhancement in irradiance as you go deeper. In contrast, when you look at a normal hydrogel, you can see this attenuation. So we then did all sorts of performance testing using some commercially relevant strains that have high lipid production. And what you can see here are these artificial tentacles that grow these microalgal aggregates of Marinochlorella. And you can see here the chlorophyll A fluorescence. And they're healthy, they're living over a time period of about 2 weeks. You can see the JAMA scaffold and these cells that grow as aggregates like grape-like structures, basically. So you have aggregates that are about, so here is some confocal, about 30 to 50 micrometer in size, rather spherical. And here you can see this growth enhancement. So if you compare this to a liquid culture, and then compare the performance to our bionic coral, have about up to 100 times higher cell densities than liquid culture. If you do light measurements over time, what is nice to see is that even at day 12, when you have the highest microalgal density, you still have sufficient light. So it only goes down to about 50% of the incident irradiance. In terms of photosynthetic performance, they're doing well, but we still have to optimize a few things with regard to the depth response. I'm not going into much detail here. And if we extrapolate this, so now trying to compare a bit with commercial techniques and approaches, in a simple open pond raceway, you can get about 4 to 20 grams per square meter per day. This is about 20 to 30 centimeters of optical depth. In our system, which is just a few millimeters, you get 2.55 grams per square meter per day, which uses about 100 to 150 times lower water volume, and 100 to 50 times lower volume and lower water usage. And of course, there's no mixing needed. So there's a few applications for basic coral reef science. We've created a synthetic microenvironment to study also coral algal interactions and algae microbe interactions. And this is of relevance for basic coral stress and physiology studies. And for bioenergy and bioproducts, of course, there's lots of interest in creating a spatial-efficient platform. So this is still your topic at this stage, but maybe there's an application in dense urban areas or as well in life support systems for space travel. But this is just the first start, really. So I'm going to thank my lab very much, the Vignolini lab at Cambridge University, and Chen lab at Nanoengineering at UCSD, as well as the various labs at Scripps Institution of Oceanography who have helped with this work, as well as the funding from the European Union, Horizon 2020, Marie Curie Actions. And if you want to contact me, please send me an email here. This is the webpage of our project, which is now concluded. And this is the web page, which is still under development of our social enterprise, where we're trying to use some new alga cultivation techniques to restore coral reefs. That's the citation to the key paper. And thank you very much.